All right, let's dive right into this explainer. Today, we're tracing an absolutely fascinating 100-year scientific detective story about how humanity completely flipped its understanding of cellular death. For the longest time, we just assumed the dying cell was a random biological accident, just a bit of bad luck, really. But as we're going to see, science has uncovered that it's actually part of an incredibly complex, highly regulated network. We're going on a journey from just staring at dead cells through a microscope to literally decoding the master systems that govern life and death. So, to really wrap our heads around this massive shift, we have to start with what seems like a total contradiction. Back in the first half of the 20th century, embryologists were peering through their microscopes watching brand new life form, but they kept spotting something super weird. Right in the exact same spots where new cells were dividing at warp speed to build an embryo, other cells were actively dying. I mean, why on earth would a healthy growing organism purposely kill off its own cells? Well, in 1951, a developmental biologist named Alfred Glucksmann hit the nail on the head. He realized this cell death wasn't a mistake. It was actually a crucial way the body shaped its tissues and organs. It was literally carving out life by removing the pieces it didn't need anymore.
Now, this observation totally shattered the biological status quo. Up until then, death was only seen as something called necrosis, and necrosis was viewed as this messy, uncontrolled disaster caused by toxins or injuries or a sudden loss of blood. Basically, if a cell died, it meant something had gone terribly, terribly wrong. But Glucksmann's work brought up a radical and honestly kind of unsettling idea. What if cell death isn't always a disease? What if it's actually a fundamental, heavily designed feature of a perfectly healthy organism?
This idea really started catching fire in the 1960s, and it came from a totally unexpected place, the silk moth. Scientists Richard Lockshin and Carol Williams were studying how these moths undergo metamorphosis, and they noticed that certain muscles the insect only needed while it was still a pupa, were degrading at highly specific, incredibly predictable times after the moth matured. This wasn't some random injury, it was scheduled. They actually coined the term programmed cell death, and suddenly a light bulb went off in the scientific community. If there's a program, there absolutely has to be a biological execution device hiding somewhere inside the cell. That hunt for the program led to our next massive leap. In 1971, an Australian pathologist named John Kerr noticed cells dying in a very peculiar way. Instead of swelling up and popping like your typical necrotic cells, these cells were shrinking. They were literally condensing their nuclei and neatly packing themselves up into small little fragments to be safely cleaned up by their neighbors. In 1972, Kerr teamed up with Andrew Wiley and Alastair Curry to officially name this active shrinking death apoptosis, and they proved it wasn't just some weird quirk. It's a basic biological phenomenon that constantly balances out cell division to keep our tissues exactly the right size.
Okay, so once apoptosis was firmly established as an active, intentional process, researchers were staring down a glaring new mystery. If a cell is actively programming its own demise, where exactly is the code written? How does a cell actually know how to pull the plug on itself?
The answer to that came from an unlikely hero, a tiny little nematode worm called C. elegans. Back in the 1990s, geneticists were mapping out the development of this worm, and they found that specific cells died at the exact same time in every single worm. It was flawless. Soon enough, they discovered the actual death genes controlling this, genes named SEED3, SEED4, and SEED9. This was huge. It definitively proved that death is governed by our DNA. Shortly after, scientists mapped out the equivalent pathways in mammals, discovering executioner proteins called caspases along with the BCL2 family. This breakthrough completely cemented cell death as a genetic and molecular process, and it even won a Nobel Prize in 2002.
For a hot minute after that, biology textbooks were wonderfully, perfectly simple. Biologists got incredibly comfortable dividing the entire cellular world into a very neat binary. On one side, you had apoptosis, the clean, regulated, shrinking suicide. And on the other side, you had necrosis, the messy, unregulated, exploding biological accident. It was clean, it made total sense.
But science rarely stays neat and tidy for long. Right when we thought we had the fundamental rules of life and death completely figured out, a massive plot twist hit. It totally shook up those clean categories and kicked off a second major revolution in cell death biology.
Researchers started noticing that some cells were dying in a way that looked exactly like that messy, accidental necrosis. I'm talking swelling, bursting, the whole shebang. But when they looked a little closer, that explosion was actually being carefully orchestrated by specific molecular signals. They named this necroptosis.
And this changed everything, because it meant you literally cannot judge a dying cell just by how it looks. Even a totally chaotic-looking death could actually be a highly regulated inside job.
Fast forward into the 21st century, and that strict apoptosis versus necrosis binary completely out the window. We saw an absolute explosion of newly discovered cell death pathways. Scientists were identifying processes with wild names like Ferroptosis, Parthenotos, and autophagy-dependent cell death. Turns out the cellular executioner system is far more intricate, diverse, and specialized than anyone back in the 20th century could have ever dreamed of.
A perfect example of this crazy complexity is a concept introduced in 2012 called Ferroptosis. This one totally broke the mold. See, Ferroptosis doesn't even rely on those traditional caspase executioner proteins that drive apoptosis. Instead, it's deeply tied into a cell's metabolism. It gets triggered by iron, a toxic buildup of damaged fats called lipid peroxidation, and a total collapse of the cell's antioxidant defenses. It proved to us that cell death isn't just a simple genetic switch, it's heavily dependent on a cell's chemical balance and metabolism.
So, if we take a step back, this entire 100-year history really boils down to a three-step evolution. First, scientists were just observing morphology, how dead cells looked, whether they swelled up or shrank down. Second, they dug deeper to find the exact molecular mechanisms, the genes, the caspases, the iron, that actually pulled the trigger. And finally, today, we're at step 3, realizing that absolutely none of these pathways act alone. They form a vast, massively interconnected cell death network.
This profound shift from just looking at the shape of a cell to really understanding its internal mechanics is exactly why modern scientific bodies completely rewrote the rulebook. In 2018, official guidelines were put in place mandating that cell death must now be classified based on its internal molecular mechanics, not just on its outward appearance.
Because of this new perspective, the questions researchers ask today are so much more sophisticated. Instead of just asking a simple, did it die from A or B, scientists are asking systems-level questions. They want to know, what exact environmental stress altered which internal molecular network to force that cell down one specific death pathway over another?
And they have to ask it this way because these systems are incredibly fluid. Key hubs like the mitochondria, calcium and iron metabolism, and lipid levels are constantly intersecting. It's like traffic in a busy city. If you try to save a cell by blocking one death route with a drug, the underlying stress might just detour right into another intersecting pathway, triggering a completely different type of death altogether.
Now, you might be sitting there wondering, okay, but why should we care so deeply about the precise molecular rules of how cells die? Why does this matter outside of a biology textbook?
Well, it matters because understanding this network is quite literally a matter of life and death for us. Almost all major diseases basically boil down to an imbalance in this exact network. In cancer, harmful cells figure out how to resist these death pathways. They just refuse to die. But in neurodegenerative diseases, the exact opposite happens, and vital brain cells are prematurely lost. In strokes or heart attacks, the sudden loss and return of blood flow triggers multiple overlapping death pathways all at once. If we want to treat these conditions, we have to know exactly how to manipulate this network to either force rogue cells to self-destruct or rescue our healthy cells from the brink.
Looking back over the last century, it's just wild to see how this knowledge evolved. We started in 1951 realizing death was just part of normal development. Then in the 60s and 70s, we coined concepts like programmed cell death and apoptosis. The 90s and that 2002 Nobel Prize proved the genetic machinery was real. And now, ever since discoveries like Ferroptosis in 2012, we are firmly in the network era. We've officially gone from viewing cell death as a chaotic biological accident to understanding it as a master control system that governs the very balance of life itself.
But before we wrap up this explainer, this incredible history leaves us with a pretty provocative question. We throw around terms like programmed and regulated all the time now. But are they really the same thing? Is every regulated death like Ferroptosis truly a programmed part of our development the same way apoptosis is? Deciphering those subtle differences in this crazy complex web of death is the next great frontier. And it's a puzzle we absolutely have to solve if we want to truly decode the language of biology. Thanks for exploring this with me and keep questioning what you see.